This is at the same time very interesting and very exceptional subject. So the story begins like that. So in exceptional cases of children with acute lymphoblastic leukemia of the B-cell lineage, they presented with clinical signs which reminds you of extremely high count of eosinophils. And indeed, some of the children comes with lung disease because of extremely high counts of eosinophils. They come sometimes with neurological disease linked to that. And sometimes the eosinophilia precede the leukemia. Anyway, when you are facing an extremely high count of eosinophils, sometimes it's not so easy to make the correct diagnosis of childhood ALL, because in these children, for reasons which are not completely well known, the count of blasts is pretty low, whatever in blood and also the marrow. Anyway, let's imagine you make the diagnosis of childhood ALL of the B-cell lineage with high blood cell count for eosinophil. One possible etiology is this translocation involving the IGH locus on chromosome 14 and the IL-3 locus on chromosome 3. What happens probably with and this has been demonstrated with this translocation, that the IL-3 gene passes under the control of the promoter of the IGH gene, which is extremely active in cells of the B lineage. And so these cells begins to synthesize IL-3, which is one of the interleukins which go towards the production of eosinophils. For example, we know that because many years ago, we tried a treatment with IL-3 for patients with black van diamond anemia, and the result of this trial was negative in terms of production of red cells, but it was positive, if I can say that, in terms of production of eosinophils. So we know that this translocation is cause of the eosinophilia. So the topic of our paper was to try to gather these exceptional cases, because the frequency is well below 1 out of thousands. It took us many years to gather the cases. We have 8 cases in France, and we also have gathered some cases from Austria. So it's totally exceptional. But the question was, besides being exceptional, what does it look like? And what we observed was, first, an extremely important male predominance. Second, we observed that the blood cell count was pretty low in most of the children in terms of white counts and blasts particularly. Third, that there were clinical signs of eosinophilia in most of the cases, and that the prognosis, which of course is of interest for many readers, was probably not as severe as could be anticipated, because some children and adolescents have been cured. Talking about adolescent, this disease has a middle age which is more towards adolescence than young childhood. And also we have had young adults with that translocation. And also interestingly, we have found some, by looking at molecular data with MLPA and other techniques, that the Icarus deletion was associated to a high proportion of this case. So the idea of the paper is to say that probably we can say that the IGH-IL3 ALL is belonging to the more prevalent but still rare group of IGH involving locus leukemia of the B-cell lineage, which are more seen in adolescent and young adults, and where the partner of IGH can be multiple, but when the partner is, and again it's exceptional, when it's IL-3, then you deal with a very special problem, because you deal with the complication of eosinophilia. Just to tell you the story of our first patient, these patients arrived with signs of CNS leukostasis, and after the beginning of chemotherapy, we have seen aggravation with coma and hemiplegia. Fortunately, the child recovered nearly completely of all these, and now we are at 7 years from that. He received an intensive treatment for chemotherapy. The MRD was evaluable at 2 time points after induction without any detectable disease. And this child has been treated for 2 years, and now he's approximately 7 years from that, and he's doing well. He has a very mild sequelae of the neurological accident, the initial accident. So it's very important for the knowledge of rare leukemias that we have international collaboration on these rare cases to understand more clearly the clinical features, like we try to assess with our data and the literature, and finally also to look more in detail at the biology, because we can imagine that in some years from now, we will try to design targeted agents to counteract the transcriptomic defects which are linked to the IGH locus, which overexpress many genes probably.